Structure and function of the hematologic system. Blood is composed of approximately 91% water and 9% solutes. In adults, the total blood volume is about 6 quarts or 5.5 liters. Blood serves several critical functions. It delivers substances needed for cellular metabolism, removes waste products, defends against microorganisms and injury, and maintains acid-base balance within the body. Plasma makes up about 50 to 55% of the blood's total volume. It is the liquid portion of blood that contains a mixture of organic and inorganic elements. Plasma proteins perform essential roles. Albumin functions as a carrier protein and helps maintain plasma oncotic pressure, while globulins serve as carrier proteins and include the immunoglobulins. Clotting factors, primarily fibrinogen, are also found in plasma and are crucial for blood coagulation. Plasma makes up about 50 to 55% of the blood's total volume. It is the liquid portion of blood that contains a mixture of organic and inorganic elements. Plasma proteins perform essential roles. Albumin functions as a carrier protein and helps maintain plasma oncotic pressure, while globulins serve as carrier proteins and include the immunoglobulins. Clotting factors, primarily fibrinogen, are also found in plasma and are crucial for blood coagulation. Leukocytes, or white blood cells, defend the body against infection and help remove cellular debris. While they are transported by the blood, they primarily act within tissues. Leukocytes are classified by structure into two groups: granulocytes and agranulocytes. Granulocytes are white blood cells that contain membrane-bound granules filled with enzymes capable of destroying microorganisms. These cells are involved in inflammatory and immune responses and are capable of amboid movement, allowing them to migrate through vessel walls into tissues, a process called diapedesis. The main types of granulocytes include neutrophils, eosinophils, and basophils. Neutrophils, also called polymorphonuclear neutrophils, are the primary phagocytes during early inflammation. Eosinophils help ingest antigen-antibody complexes and are particularly important in IgE-mediated allergic reactions and in defense against parasitic infections. Basophils are similar to mast cells and are involved in allergic and inflammatory reactions. Agranulocytes include monocytes and lymphocytes. Monocytes are immature macrophages that, once matured, become part of the mononuclear phagocyte system. Lymphocytes mature into T cells, B cells, and plasma cells, playing critical roles in adaptive immunity. Natural killer cells, or NK cells, are a distinct type of lymphocyte that targets virus-infected and abnormal cells. Platelets, also called thrombocytes, are irregularly shaped cytoplasmic fragments essential for blood clotting. They play a critical role in coagulation and in controlling bleeding following injury. This final composition slide references key structural illustrations of blood from leading histology and anatomy texts. Together, plasma, erythrocytes, leukocytes, and platelets work harmoniously to support health, protect against disease, and maintain homeostasis. The spleen is the largest lymphoid organ in the body. During fetal development, it plays an important role in blood formation, known as hematopoiesis. In adults, the spleen filters and cleanses the blood, initiates immune responses, and removes aged or damaged blood cells. It also serves as a reservoir, storing approximately 300 milliliters of blood. Within the splenic pulp contains masses of lymphoid tissue, rich in macrophages and lymphocytes, while venous sinuses are sites where old blood cells are phagocytosed and destroyed. Lymph nodes are small, bean-shaped structures that are part of both the lymphatic and immune systems. Structurally, they filter lymphatic fluid before it is returned to the bloodstream. Functionally, Lymph nodes cleanse the lymph of microorganisms and foreign particles. They are strategically located throughout the body to monitor for signs of infection and to help mount an effective immune response. This diagram highlights the internal structure of a lymph node. Inside, lymph flows through a network where it encounters immune cells, such as lymphocytes and macrophages, that work to detect and eliminate pathogens. Lymph nodes are essential for both immune surveillance and activation. Blood cell production, or hematopoiesis, begins in the bone marrow. Red or active marrow is hematopoietic and is found primarily in flat bones in adults, such as the pelvis and sternum. It is highly vascularized to support cell growth. Yellow marrow, in contrast, is inactive and consists largely of fat. Under certain conditions, yellow marrow can revert back to red marrow to meet increased demands for blood cells. Within the bone marrow, special microenvironments called niches support the development of blood cells. Mesenchymal stem cells form the stromal framework and can differentiate into various connective tissue types. Hematopoietic stem cells give rise to all blood cell types. These cells are regulated by colony-stimulating factors, which guide their differentiation into specific blood components. Cellular differentiation begins with multipotent stem cells that have the potential to become any type of blood cell. These cells then further specialize into progenitor cells that are committed to becoming a particular lineage, such as erythrocytes, leukocytes, or platelets. Hematopoietic cells are self-renewing and responsive to growth factors like erythropoietin, which stimulates red blood cell production. Hematopoiesis is the ongoing process of blood cell production. In the fetus, hematopoiesis occurs mainly in the liver and spleen. After birth, it shifts to the bone marrow. Hematopoiesis involves the simultaneous proliferation and differentiation of blood cells into their mature forms. Occasionally, blood cell production can also occur outside the bone marrow in a process called extramedullary hematopoiesis, which typically signals disease. This diagram shows the differentiation pathways of hematopoietic stem cells into various mature blood cells. Each branch represents a stepwise process influenced by growth factors, leading to the production of erythrocytes, granulocytes, monocytes, lymphocytes, and platelets. This careful regulation ensures that the body maintains proper levels of all blood components. Erythropoiesis is the process of red blood cell development. It follows a sequence beginning with progenitor cells, then progressing through stages as proerythroblasts, erythroblasts, normoblasts, reticulocytes, and finally mature erythrocytes. At each step, the amount of hemoglobin inside the cell increases, while the nucleus gradually shrinks. Erythropoiesis is regulated by a feedback loop involving erythropoietin, a hormone that stimulates red blood cell production in response to tissue hypoxia. This visual reinforces the stages of erythropoiesis. Erythropoietin, produced mainly by the kidneys, acts on the bone marrow to promote the formation of new red blood cells, particularly when oxygen levels in the blood fall. This tight regulation ensures the body can adapt to changing oxygen demands. Proper red blood cell production depends on specific nutritional requirements. Key proteins and amino acids are needed, along with a wide range of vitamins, including B12, B6, B2, folic acid, pantothenic acid, niacin, vitamin C, and vitamin E. Essential minerals such as iron and copper also support hemoglobin synthesis and overall red blood cell health. As erythrocytes age, they lose their flexibility and become increasingly fragile. Old or senescent, erythrocytes are recognized and removed by macrophages, primarily in the spleen. If the spleen is dysfunctional or absent, the liver takes over this role. The normal destruction and recycling of aged red cells ensure that iron and other components are conserved and reused. Hemoglobin is the oxygen-carrying protein found inside red blood cells. It consists of two pairs of polypeptide chains, usually two alpha and two beta chains in adults, and four heme groups. Each heme group contains an iron atom that binds to one oxygen molecule. For oxygen to bind properly, iron must be in its reduced ferrous form. If iron is oxidized into the ferric form, it forms methemoglobin, which cannot carry oxygen. The precise arrangement of globin chains in heme groups allows hemoglobin to efficiently bind, transport, and release oxygen where it is needed. Disruptions in hemoglobin structure or iron availability can severely impair oxygen delivery to tissues, resulting in clinical conditions such as anemia. Iron is a critical component of hemoglobin and is carefully regulated in the body. Most iron is either bound to heme within blood and muscle cells or stored as ferritin or hemosiderin within macrophages. Only a small amount is lost daily through urine, sweat, bile, or skin shedding. To conserve iron, the body recycles it from aging erythrocytes, a process tightly controlled by the hormone hepcidin. This diagram shows how iron is reclaimed from destroyed red blood cells and reused for new hemoglobin synthesis. Maintaining the balance between iron absorption, storage, and recycling is critical for sustaining healthy red blood cell production and preventing disorders such as iron deficiency anemia. Leukocytes, or white blood cells, develop through a process called myelopoiesis. Granulocytes and monocytes mature in the bone marrow. Granulocytes are divided into two pools, a circulating pool in the bloodstream and a marginating pool stored along the walls of blood vessels, ready for rapid deployment. Lymphopoiesis describes the development of lymphocytes, which are released into the blood and mature in lymphoid organs such as the spleen and lymph nodes. Platelets, or thrombocytes, are produced through thrombopoiesis. In this process, a large cell called a megakaryocyte replicates its DNA multiple times without cell division. Portions of the megakaryocyte cytoplasm break off as platelets. Each megakaryocyte can produce thousands of platelets. Platelet production is regulated by thrombopoietin, and platelets circulate for about 10 days before being removed by the spleen. Hemostasis is the process that stops bleeding through the formation of a blood clot. It begins with vasoconstriction in response to vascular injury. Endothelial cells at the site of damage release signals that cause platelets to adhere to the exposed surface, forming a temporary plug. Simultaneously, the coagulation system is activated, producing fibrin strands that reinforce the platelet plug. As the clot matures, it contracts to form a more permanent seal, preventing further blood loss. The first component of hemostasis involves the blood vessels themselves. Endothelial cells lining healthy blood vessels help maintain fluidity by producing nitric oxide and prostacyclin. When damaged, these cells expose the subendothelial matrix and release activators like von Willebrand factor, which initiate clotting. This local response helps control bleeding while preserving circulation in surrounding tissues. Platelets play a central role in hemostasis. They regulate blood flow by promoting vasoconstriction, form the initial platelet plug, and activate the coagulation cascade to stabilize the plug with fibrin. When a vessel is damaged, platelets adhere to the exposed surface, become activated, release their contents through degranulation, and then aggregate to form a dense cluster. They also help initiate the repair process by aiding in clot retraction and dissolution. Clotting factors create the fibrin mesh that stabilizes the platelet plug. These factors operate through three pathways. The intrinsic pathway is triggered when factor XII contacts exposed subendothelial tissue. The extrinsic pathway begins when tissue thromboplastin is released from injured endothelial cells. Both pathways converge at the common pathway, which activates factor X and ultimately results in the formation of a stable blood clot. This diagram depicts the clotting cascade in detail, showing how the intrinsic, extrinsic, and common pathways interact to activate thrombin and convert fibrinogen into fibrin. The cascade ensures that clotting occurs precisely where it's needed, and the presence of multiple feedback loops amplifies the response to injury while maintaining regulation. The body has built-in checks to prevent spontaneous or excessive clotting. Endothelial cells release thrombin inhibitors such as antithrombin, which neutralizes active clotting factors. Tissue factor pathway inhibitor blocks the initiation of clotting. Protein C helps degrade activated clotting factors. These control systems maintain a balance between coagulation and anticoagulation to protect against both hemorrhage and thrombosis. After a clot forms, it undergoes retraction, during which fibrin strands shorten and pull the edges of the wound together. This process is facilitated by the platelets within the clot. Once healing has progressed, the clot must be broken down. This is achieved through the fibrinolytic system. Plasminogen is converted to plasmin, which digests fibrin and dissolves the clot. The resulting fragments, called fibrin degradation products, are cleared from the circulation. The fibrinolytic system is responsible for breaking down clots once they've served their purpose. Plasminogen, an inactive precursor, is converted into plasmin by tissue plasminogen activator. Plasmin then degrades the fibrin mesh into soluble fragments. This system helps restore normal blood flow and prevents clots from persisting too long or forming where they aren't needed. At birth, blood cell counts are elevated compared to adult levels. This increase is due to the physiological stress of delivery and the cutting of the umbilical cord. Additionally, the relatively low oxygen levels in the womb stimulate fetal erythropoietin production, leading to a higher number of red blood cells, a condition called polycythemia. This adaptation helps ensure oxygen delivery during the early transition to life outside the uterus. Newborns are at increased risk for infection due to immature immune function. Their ability to perform phagocytosis is limited, which affects their response to bacterial threats. Wound healing is also slower during this stage. Fortunately, maternal antibodies, specifically IgG, are transferred across the placenta and provide temporary passive immunity. This early protection is crucial until the infant's immune system fully matures. As people age, their hematologic system gradually declines in function. Although the lifespan of red blood cells remains normal, their production slows due to diminished iron absorption and reduced erythropoietin response. This often leads to mild anemia in elderly individuals. Additionally, lymphocyte function declines, impairing the immune response. The humoral immune system also becomes less responsive, increasing susceptibility to infections and reducing vaccine effectiveness.